So in fact, this CN business, there's a plane coming out of my arm here. And likewise, there's another plane. So that I'm already sort of indicating some of the geometric rigidity. Furthermore, at a C alpha, these are tetrahedral angles. So there are these two planes coming in with the conformational angles phi and psi along the backbone, and you know, my humpback and all that are tetrahedral. So I'm sorry, I sort of jumped the gun. I finished the bad poetry, and I described the chemical, and we've moved on already to the geometrical. Let me write down some of the things I just said, because the profound fact, we move now on to the, just make sure I don't forget, because there will be some things we need to, no, that's good. Some profound things we'll need later. So geometrically, now moving on to the third of the bullet point for proteins. First of all, I said it quickly, but this picture is accurate. There's a carbon and then the next nitrogen along in the next amino acid. There's the C alpha and the C alpha, and then there's remaining from the hydration of the water molecule an O and an H. And I'm putting sort of partial double bonds in here for reasons that I'll now explain. There's a miracle here, and indeed a miracle of quantum mechanics. The miracle is that this is planar. These 1, 2, 3, 4, 5, 6 atoms, by which I mean the center of the Bohr model, all lie in a plane. And this is truly, obviously, to a mathematician, a miracle. So the miracle is this, it's called a peptide unit, a sub-molecule of a polypeptide consisting of these 6 atoms. This peptide unit is planar. And this comes from hybridization, an sp2, sp3 hybridization of bonding here that forces these guys to be in a plane. And then there is some further hybridization involving also the electrons on this oxygen that really rigidify the entire gadget. So not only is the peptide unit planar, but furthermore, these angles are very closely 120 degrees. Jorn and I have gone through the database, which I'll describe in a bit, and these are really quite accurate facts. Of course, we're doing real life, somehow science, so everything has some experimental error and blah, 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 but these peptide units are really planar to as many digits, I forget how many digits we sort of looked at, but to a huge number of digits. This is really true, not just an approximation, and it's a miracle. So you see, these proteins are really quite rigid. There's this plane, and then tetrahedral angle, and then a plane, and so on, and the pattern repeats. So this is sort of the rigidity of the story. Maybe there is a tiny amount more to say, well, as you, yeah, go ahead.
The elastic in some sense, if you bump it by some other molecule, will they bend?
Well,
dynamics there?
We'll come to the dynamics, and they better not bend very much, or you're dead. So, well, no, I'm serious. We'll come to...
No, I bend in the sense that so elastically bend.
This fact, you're meaning elastically.
Yeah.
Again, in our experience with the data, these are, the 120 degrees, there is some variation, but there is some experimental error in the data, but the planarity is just dead on. We really checked this in some detail over 25 million of these peptide units, and it's dead on. So no, this is really on the nose. Maybe there, I should just mention, it won't play a role in our model or anything, but there's also a chirality. That as you look from my hydrogen hat down at the bonding, it goes carbon residue nitrogen, as in this figure. So there's a chirality in proteins, at least as they are synthesized. Just that. All right. So I think that's what I wanted to say geometrically. So I'll continue coming up here and checking things off so we can keep track of ourselves. There's the geometrical discussion. The dynamical discussion, maybe I'll move over here. The dynamical discussion is really fascinating. And sort of in the spirit of the answer that I gave to what I thought Koya was just asking. So, dynamically, kind of again, another miracle. And one of the fundamental parts of the story is that proteins, let's say, use the vernacular, the usual biology vernacular, fold into, there are going to be too many quotes here for your liking and mine as well, fold into crystals. Now, I don't mean crystal in any precise mathematical or physical sense. I don't even mean a quasi-crystal in any precise mathematical sense. What I mean is an object where one can quantify this by saying, and make precise now the definition of protein. A protein, I give the definition, is a linear polymer of amino acids that folds into a crystal. Now I have to tell you what I mean by a crystal, to be precise. And what I mean is that it saturates space, so it sits in some ball, and 80% of that ball, say, is occupied by the atoms of the, we choose some percentage, 80% let's say, is occupied by the atoms in the protein. So I mean by crystal partly that it folds into some compact, hard shape. Now what do I mean by hard? Hard is that the lowest energy state is well separated from the nearest competitors. So this thing, this protein folds into a crystal, and it's hard. Now there's not much elasticity. This is what I'm saying. You bang it a little, it ain't gonna move because the nearest energy states are pretty far away. And again, it must be so in order to function properly. Maybe I'll discuss the functioning presently. But go ahead, Clay.
And this separation, this ground state is separated.
Yes.
Excited state. Is it quantum mechanical separation or is it classical sort of energies?
I'm not sure exactly how to answer this.
I mean, this
How do you mean?
this energy,
Yes.
measured in Planck constants. Is it something microscopic?
No, I see. The typical, okay, I need to give you some, in order to put this, let me answer the question and then put it in some perspective. It's sort of 10 kilocalories per mole is the amount of energy, the stability region for a typical protein, 10 kilocalories per mole. But that's not in much context until I tell you some of the other energetics, which I'll do in a moment. I mean, remind me and I'll tell you what these other effects cost relative. But 10 kilocalories per mole is like a reasonable number of stability.
But there is no continuous spectrum in the system.
No.
That means it's not quantum.
Again, in principle, it is quantum mechanics. After all,
But
it's a
that's
bunch of atoms. But the problem, we can't even do helium. So to consider doing a linear polymer like this, or even a single amino acid, is out of the question. You have a comment?
Well, no,
Please feel free to ask something.
I think these are experimental numbers which are sort of independent of
Oh,
what
to be
theory
sure.
we throw at it, right? And so the discussion about whether it's quantum or classical is a little bit...
There is an evolved discussion in the biology community as to what extent should quantum mechanics be part of understanding this folding process, and nobody really knows. This appearance of quantum mechanics to make the peptide units planar is as far as I think we can reasonably go here, quantum mechanical. Okay, so, and then finally, as a mathematician, there I think is the definition. A protein, let me write it, because this is, a protein is a linear polymer of amino acids, that folds into a crystal. There are experiments to show that among all polymers, the proteins are extremely rare. I mean, 1 in 10 billion of random chains of residues glued together actually folds into a well-defined crystal in the sense that I gave this definition. So these are extremely rare and have been biologically selected in order to produce life as we know it. Okay, so here is the, I guess there are now a number of things to say. One is, you may worry that, well, perhaps the cell, the organism, actually takes this protein and folds it as part of the mechanism of the cell. In other words, do proteins self-organize? Do they fold on their own? Or does something in the cell have to organize, in some organized way, fold them? And the fact that has been, that goes back to the 60s. Herein lies a Nobel Prize.